Here at the Vasculitis Expertise Center in Groningen, I see many patients with GCA and PMR. And all these patients have two important questions. The first question is, why did I develop GCA or PMR? What is the cause of these diseases? And the second important question is, do I really need prednisolone? Are there any better alternatives to prednisolone? Because the prednisolone has many side effects. So in my research, I try to answer these two questions. Regarding the first question, what is the cause of GCA and PMR? I believe that it's very important to look not only in blood in these patients, but also and especially at the site of inflammation, being either the arteries of patients with GCA, both the temporal artery and the aorta, and the bursae, especially the shoulder bursae in patients with PMR. I am especially interested in inflammatory cells in GCA and PMR. And during the last years, we, together with Professor Brouwer, we focused on two types of cells. The first, monocytes and macrophages. These are a type of inflammatory cells which we know they are important in GCA. But for instance, we recently found that a certain type of macrophages, a more aggressive type of macrophages, the so-called CD206 positive macrophages, you can see them here in brown and over here in green. So these are the aggressive macrophages. They are able to produce a protein which is called MMP9. And this protein is able to degrade the vessel so that the inflammatory cells can easier get further into the vessel wall and induce more damage. So we believe that this type of macrophages may represent a new target of intervention of therapy in GCA patients. What we also have found recently is that another protein, which is called GM-CSF, and that is a protein important for the growth and also for the function of monocyte macrophages, is also expressed in, at the site of inflammation in GCA. And as a matter of fact, there was a recent study with an antibody called mavrilimumab, which blocks the receptor of the GM-CSF, which looks promising in this treatment study. I am personally very interested also in another type of inflammatory cells, the B cells. And the B cells are well known for the fact that they are the precursors of the plasma cells, and the plasma cells produce antibodies. Which is very interesting because the B cells we have recently showed that the B cells are massively present in the aorta of patients with GCA, but also in the temporal artery. So they are present at the site of inflammation. However, there are no antibodies, specific autoantibodies found so far in patients with GCA. So we believe that the function and the role of B cells in GCA is more related to their capacity of producing inflammatory proteins, cytokines. So that we believe that the B cells may represent another future interesting target of intervention in GCA. I am also interested not only in inflammatory cells, but also in the behavior and the role of residing cells, so the cells which are normally present in arteries. For instance, fibroblasts, what are their role, how do they behave, and what is their function and role in inducing more damage in the arteries in these patients. Because as you may know, the arteries of patients with GCA can get either narrowed and even obstructed, or that they can get dilated, for instance, the aorta. We also do more recently studies, DNA and RNA studies in blood and in tissue. The goal of these studies is to learn about the mechanism of diseases in GCA and PMR so that we are able to better treat the patients. So the ultimate goal of our research, both in understanding and treatment studies in the GCA and PMR, is to be able to better treat these patients so that their quality of life will be much better than it is nowadays. I thank you for your attention.